Good evening, all. Hello. Good evening, all. Yesterday we completed protein synthesis and auxins physiological activity. So in phytohormones, as we discussed in last class, auxins, gibberellins, cytokinins, abscisic acid, ethylene. Right? So in last class, auxin physiological activity completed. Am I audible?
Yes, sir.
Hello?
Yes, sir.
Yes, sir.
Right. Right. Now gibberellin and gibberellic formation. Open your book, all of you.
Sir, page number?
I don't know. I have no book. That's why I'm saying it.
Okay.
Gibberellin, which is extracted from the rice plant when it is infected with foolish disease, rice foolish disease, which is caused by Gibberella fusicorae. Right? So during the infection of Gibberella fusicorae infection, when the scientists find out the hormone, which is actually the foolish disease, the pale and elongated, and it is appear very thin in structure, and very thin and elongated structure, and almost reproductive parts are destroyed and down, and this is called foolish disease. And first the scientist, I think Ebuta or Kurosawa. First extract who is Kurosawa, I think. Hello?
Yes, sir.
Who is the first scientist extracted gibberellin? See in book. Kurosawa. Right, Kurosawa. Actually Kurosawa extracted but not given any name. Just he find out as a phytohormone from the gibberellin fusicorae. But later the name given as a gibberellin Ebuta. The name given as a gibberellin by Ebuta. Right? So this is the small introduction about the gibberellin. And now the physiological activity. What are the physiological activity given in our book? I think first one, elongation.
Yes, sir.
Second, bolting and flowering. Right?
Sir.
And third one, seed dormancy removal. Hello? What are the physiological activity? First, elongation. Second one, I think rosette. Sorry. Second, what is that?
Elongation, growth,
Bolting.
and bloating.
Bolting. Right.
Yes.
Bolting and flowering. And third one, seed dormancy.
Yes, sir.
And fourth one, parthenocarpy and sex determination. Right? First,
Sir.
let's discuss the first one, the elongation. So actually, the main effect of the gibberellins, when it is less in constant, the almost stunted growth. But when we apply the externally, it shows a rapid growth of the plant. Let's see here, just wait. Let me share the screen. Are you able to see this?
Yes, sir.
Hello?
Yes. Yes, sir.
Right. Actually, this is the first one.
Yes.
We could not find out the nodes and internodes in A diagram. When we apply the external is the gibberellic phytohormone, due to the effect of gibberellin hormone, these nodes and internodes rapidly increased and appeared. Just wait. Right. So this is the first plant without gibberellic reaction. But in B diagram, when we externally apply the gibberellin, actually in this first diagram, here the nodes, this is node, this is internode, and again node. These nodes and internodes compactly arranged in first diagram. That's why we are unable to see the nodes and internodes. But what is the next diagram, B diagram? In B diagram, we can see clearly the nodes. These are the clear nodes. Clear nodes are clearly, that means the node from the every node region, the leaves are arranged. For example, this is node, this is node, this is node. Produce the leaf, leaf, leaf. This is after application of the gibberellin. But before application, actually, this is the plant growth. The nodes and internodes, for our convenience, I had to draw like this. Actually, they are very compactly arranged like this. So every cross line is considered as a node, and between the node, that is called as internode. So this is before apply of the gibberellin. This is after apply of the external apply of the gibberellin, right? Is it clear?
Sir.
That is the first physiological effect. And second one, what is second? Bolting and flowering, right? Hello? What is the second one?
Sir. Yes, sir.
Right.
Bolting and flowering.
Right. Let me share the another diagram. Just wait. Bolting. Are you able to see this diagram? Hello?
Yes, sir.
Sir, yes, sir.
Right. Bolting and flowering in mainly in Brassicaceae family, such as the best example mustard. In what happened in Brassicaceae family? Before flowering, actually during vegetative state, there is no production of gibberellin. That's why in first stage, before flowering stage, only the leaves appear as a rosette like this. So this is only vegetative. And this type of arrangement called as rosette formation. The leaves in arranged in rosette, because in rosette means actually, for this is the stem, this is leaf, right? This is leaf, right? And is somewhere here also leaf, right? But we can clearly visible the node and leaf structure, node and leaf structure, node and leaf structure. But this is, we can see only the after release of gibberellin. But before gibberellin, actually these nodes and internodes closely, compactly arranged. That means they are arranged. This is node, and again node, and again node. Due to this arrangement, the leaves also arranged as a compactly arranged, very closely arranged. So this type of arrangement called as rosette formation. This rosette formation up to vegetative stage. When the entering into the reproductive stage, immediately inside the plant body, that is not external. Inside the plant body, immediately the gibberellin phytohormone released. As a result, all of a sudden it is rapidly increased and production of flower. Actually, this is not correct. After rapidly increased, then only the flower produced. That is the indication of flowering stage. This is called bolting. Bolting means, simply say one word, the arrangement of the leaf in a rosette formation. And in vegetative stage, when it is entered reproductive, immediately the gibberellin already released inside the plant body, and nodes and internodes elongated, rapidly elongated. So this is called bolting and flowering. Is it clear? Hello?
Yes, sir.
Right. What is the third one?
Germination, sir.
Seed germination, right?
Yes, sir.
Just wait.
Dormancy.
Seed dormancy removal. Seed germination and seed dormancy. What is the heading? Are you able to see this diagram?
Yes, sir.
Right. This is the seed structure. The outer line seed coat. This is this red color. This one, this endosperm, and this portion called embryo. And this endosperm and embryo covered with the protein layer that is called alluron. And this I already told you, seed coat. This is the seed structure. So it is promote seed germination. Gibberellin promote the seed germination. Without gibberellin formation inside the embryo, there is no germination of seed, right? Let's see the mechanism of the seed germination after production of the gibberellin. Actually, the first after matured embryo, the embryo imbibes the water and swell the same, right? Imbibes the means absorb the water. What is imbibition? What is imbibition? Already told you in very beginning in offline class. Osmosis, imbibition, diffusion. What is imbibition? Imbibition. Hello?
Receiving, sir.
What? Imbibition. Imbibition, observation of the water molecule from the dead cells. But where the dead cell in seed? That means simple seed coat the cells, dead cells. So this dead cell absorb the water by imbibition process. After that, which is received by embryo, right? In second stage, what happened? The embryo secretes gibberellin. Immediately after receive the water molecule, the embryo in second step, the embryo secretes the gibberellin and diffuse into alluran layer. So actually gibberellin formed inside the embryo, later diffused into the outer layer alluran layer, where they trigger the synthesis of hydrolytic enzymes. What are the enzymes here? Alpha amylase enzyme. One is alpha amylase or proteinase enzyme. Right? This way. Right. So after formation, alpha amylase and or proteinase, anyone or both, here where the storage food material present inside the seed, storage food material in seed, embryo, endosperm, alluran layer or seed coat. Storage food material portion of the seed.
Endosperm, sir.
Endosperm, right. In endosperm, the storage food material, maybe if it is protein or carbohydrate, starch, in the form of starch or in the form of protein or in the form of fat, they are stored inside the endosperm, right? That is based on the plant species. So after release of the amylase enzyme or proteinase enzyme, this amylase already you must have heard this word amylase. Amylase enzyme acts on starch. The starch material already stored inside the endosperm. This starch convert into simple sugar. If it is protein released, these proteins convert into what are the subunits of proteins? If in the endosperm proteins are stored, after proteinase enzyme, these proteins convert into what are the simple form of the proteins? We discussed last class.
Amino acids,
Right.
sir.
Right, amino acids convert into amino acids, right? Convert into amino acids, right? So first let's discuss one enough, that is amylase. If amylase released, the starch convert into simple sugars, may the best example glucose, right? Hello? Simple sugars,
Sure.
that is best example, convert into glucose. Is it clear? Hello?
Okay, sir.
Yes, sir.
Right,
Yes,
after where the glucose now? Where is the glucose now? Actually, glucose in the endosperm. The later inside the endosperm later diffused into the embryo. Glucose after convert into that means the starch after convert into glucose, which is diffused into the embryo, where the respiration takes place. What is respiration? What are the stages of respiration? What is the end product of respiration? Already these all classes are completed. Now here the glucose substrate is clear, right? Now tell me what happened after formation glucose during respiration process. Hello? Glucose convert into
Pyruvate, sir.
right by which mechanism, which cycle?
Glycolysis, sir.
Glycolysis, right? From glucose glycolysis is which is converted into pyruvic acid, right? Next step, from pyruvic acid,
Oxidative phosphorylation.
oxidative decarboxylation, As a result, formation of coenzyme A, right?
Yes, sir.
Now this coenzyme A enter into the
Krebs cycle.
Krebs cycle. During all these stages, some direct ATP are formed or in the form of NADPH2 or FADH2 also formed. What is the last stage in respiration? What is last stage? Hello? What is the after Krebs cycle? Krebs cycle. Okay. What is the end product of respiration? Hello? End product of respiration.
ATP, sir.
ATP. How the ATPs are produced?
Through electron transport chain.
Through electron transport chain.
Transport
That is the
chain.
last cycle. So now after in glucose glycolysis or connecting link or Krebs cycle, where the formation NADPH2, FAD, they are all enter into the ETS cycle and finally produce ATP.
Yes, sir.
After ATP produced inside the embryo, then utilize this energy and germinated out. These are in general what we call sprouts. That's why these are the more energetic, right?
Yes,
Is it
sir.
clear? So this is the mechanism of germination. Okay? Is it clear?
Okay, sir. Yes, sir.
Right. The next one. What is the next heading after germination? Parthenogenesis, I think, or sex determination. So first, let me complete the sex determination. Auxins promote female flower, where the gibberellin promote male flower. That is the difference. The same mechanism, sex determination, in done by auxins and also gibberellin. But in auxins, only promote the female flower in cucumis and cannabinus, I think. Right? Cannabinus sativa, right? Cucumis and cannabinus sativa, due to effect of the auxins, only the female flowers are produced. But in gibberellin, with the effect of the gibberellin, the same plants, cucumis and sativa plant, only male flowers are produced. Staminate flower, I think, staminate or male flower, both are same. So that is the sex determination. And last one, parthenocarpy. What is parthenocarpy? Is there parthenocarpy or not? Hello? In our book given about, yeah,
Yes.
I think in
Yes,
grapes, right, in best example in grapes, parthenocarpy by gibberellin. What is parthenocarpy? Already we discussed in auxins.
Without
What is parthenocarpy?
fertilization, sir.
Without fertilization, the fruit formation. So
Fruit
there is no
formation takes.
right. There is no fusion between the male and female gamete. As a result, no formation of embryo and seed. That's why these fruits are seedless fruits. In gibberellin effect, mainly in grapes, grape plant. That is physiological activity of gibberellin. Anyone missing here? Hello? All headings are covered. Are you in book? Are you with book?
Yes, sir.
Right. First of all, elongation. Second, bolting and flowering. And next one, seed germination and sex determination, parthenocarpy. That's it, or anyone?
No, sir, that's it.
Right. Now the third one, cytokinins.
Cytokinin.
Right. Who discovered cytokinin? Is there any name?
When, sir?
Went.
I think so, sir.
Went, I think, in auxins.
Auxins, cytokinins were discovered by Johnson.
It's not given in our book.
No, sir, I am outside, sir. That's why I'm not seeing the textbook.
Just wait. Any chemical name given cytokinin? Chemical name. Right. Cytokinin discovered by Skoog, and is named as a kinetin. Right? Skoog, discovered by Skoog. If it is not given, write down the Skoog, and but the name given as a kinetin. Okay, enough. And cytokinin chemical name. Here the chemical names most important. I think oxygen chemical name IAA, indole acetic acid. What about gibberellin chemical name? Is given or not? Gibberellin chemical name. Hello?
GAA, sir.
GA3.
Yes, sir.
Right. What about the cytokinin? I think 6-furyl cytokinin chemical name. Already given, I think. Given or not? Six methyl buto 2 ethyl aminopurine. 6,4-hydroxy 3 methyl buto 2 anilaminopurine. So that is the cytokinin chemical structure. Right. Okay. Now the physiological activity. What are the physiological activity given in our book? I think cell division, cell elongation, and another one is senescence, delay senescence, right? Delay senescence. Another one in cytokinins, morphogenesis. Most important, morphogenesis. And stomatal opening. Stomata opening. Right? Apart from that, any other extra headings? Hello? Hello?
Removal of removal of apical dominance.
Apical dominance, anti-apical dominance.
Sir.
And apical dominance. Apical dominance done by which hormone involved in apical dominance?
Oxygen, sir.
Auxins. So this is anti, that is the opposite direction, opposite activity, right? Yes, so first let's discuss first one, cell division, right? Cell division, due to the effect of the cytokinin, the cells are rapidly divided and redivided and form the group of cells, which is found when the crown gall disease occurred in the bacterial name such as Agrobacterium, right? Agrobacterium tumefaciensis. When the Agrobacterium tumefaciensis infected the plants, automatically the plant roots form some galls. Galls means that is the tumor formation. That's why this is called as a gall disease. When the scientist is extracted from the gall cells, that is tumor cells, that dramatically they find out some phytohormones, which is the named as a cytokinins. That's why they confirmed the activity of cell division done by cytokinin, right? That is first one. And apart from the cell division, cell elongation also done by cytokinins. What is cell elongation? Cell division. Cell division, one cell becomes 2, 4, 8, like rapidly increased. What is cell elongation? The volume increased. Only the cell volume increase, but not number. That is cell elongation. Cell division, cell elongation, the both are done by cytokinins activity. And another one most important, delay senescence. What is senescence?
Programmed cell death, sir.
Programmed cell death.
Yes, sir.
Just read out once. In our book, what they given? Delay senescence means, okay, programmed cell death. Aging, delay aging. Delay aging means here, especially what happened in the leaf. Actually, in initial stage, the leaf are in green in color, right?
Yes, sir.
While
Early
this,
aging, sir.
why,
Yes.
right, early as in green in color, while the years are goes on, it is automatically turns into yellow in color.
Yes, sir.
The yellow represents the age,
Yes, sir.
aging. Green color, they are the first one, but later these are when enter into the old age, these leaves convert into yellow in color. So, but this activity delay by cytokinins, that is called senescence. So here, actually what happened in leaves, let me share the diagram. Okay. No diagram, just wait. Right. So actually the green color mainly, how the green color formed inside the leaf due to the activity of, due to the presence of, hello?
Chlorophyll, sir.
Chlorophyll, presence of chlorophyll. If the chlorophyll degrades or destroyed, what happen?
The color of the leaf will change, sir.
Right, color of the leaf change. That's why it turned into yellow in color. But this degradation of chlorophyll delay by cytokinins. In the presence of cytokinins, automatically there is no degradation, there is no destroy of the chlorophylls. They are always in active, and this gives always green in color. That's why this is called as delay senescence, right? That's why in the most of the florists this utilize the cytokinin, because the fresh appearance of the flowers and leaves. When we apply the cytokinins to leafy vegetables or flowers, automatically they never shut down. They are always in appearing fresh, because these cytokinins delay senescence, delay the convert into old age, right? So this is delay senescence by cytokinins. What is the next one? Audible?
Yes, sir.
Hello?
Yes, sir. Yes, sir.
Right. But what is the next heading? After delay senescence clear, or any doubt in this?
No, sir.
No, sir.
Right. What is the next heading? Morphogenesis, right? Hello?
Sir. Yes, sir.
Morphogenesis. Morpho means morphological structure. Genesis means formation. If the morphological structure means root formation and shoot formation. So the quantitatively, the ratio of the auxins and cytokinins, they form the root and shoot. Now just tell me, in morphogenesis, right. auxins, cytokinin. High auxin, low cytokinin, production of root. That is called rhizogenesis. So remember, high auxins and low cytokinin, production of root. That is called rhizogenesis. And opposite, high cytokinin, low auxin, production of stem. That is called collagenesis. So during plant tissue culture, after formation of callus, here the callus, there is, what is callus? What is callus? What is callus? Callus means undifferentiated.
Undifferentiated mass of cells, sir.
Right, undifferentiated mass of cells. But when we apply with cytokinin and auxin with ratio, if we want to get the roots, high auxin and low cytokinin ratio, they produce the root. At the same, if we use high cytokinin, low auxins, the production of stem. So due to the production of root and stem, almost the small rootlets are formed. So this is called morphogenesis, right? The morphogenesis mainly involved by cytokinins as well as the portion of the auxins also, right? Is it clear? Hello?
Yes, sir.
Right. What is the next one? Apical dormancy, right? Anti-apical dormancy. Already we discussed about the apical, what is apical dormancy during the class of axins. Apical dormancy means a linear growth. What is anti-apical of cytokinin? The growth appear like a umbrella-like a structure, not a conical structure, not a cone structure. If it is apical dominance, if it is activity of axins, it is upright direction, goes upright direction, and the plant body appear as a conifers, cones. What happened in the cytokinin activity? In cytokinin activity, the plants appear as a, is it linear growth or vertical growth? Sorry, horizontal growth. Due to the activate cytokinin activity, that means the simple opposite to the axin activity of apical dominance, right? Hello?
Yes, sir.
Right. So due to the activity of cytokinins, here the branches are formed heavily. That's why this plant body appear as an umbrella-like structure, but not a conical structure. Is it clear?
Can you repeat
Hello?
it, sir?
Right? Right. This is clear. Apical dominance clear? Hello? Hello?
Yes, sir.
Hello? Right. In cytokinin, tell me. Someone raise the doubt. Please ask me. Hello? Right, okay. If you have any doubt, ask after the class. I will give you 5 minutes later. Right. Next one. After auxin, what is the next one? After cytokinin, what is the next one? Abscisic acid, right?
Yes, sir.
Right. Abscisic acid, I think the scientist Abucart. Which scientist name given? Just open your book. Abscisic acid, right, which is discovered in cotton fruit, right? Cotton fruit. The scientist name, I think, Abucart. Which scientist given? Open your book.
Frederick T. Adicott, sir. Frederick
Who
T.
is that?
Adicott. Rani, sir.
Right, Adicott. Right, Adicott, Adicott. Right, right. Okay. Okay, next, what are the physiological activity of the abscisic acid? Actually, this is the regulatory hormone. That is, first one, the axins, gibberellin, cytokine, they are the promoters. These the promoters, and next ABA, abscisic acid, and ethylene, these two are inhibitor hormone. That means almost all opposite direction, opposite activity of the axins, gibberellin, as well as cytokine. Right? What are the physiological activity of abscisic acid? What are the first heading? Hello? Stomatal close. Sorry, one thing is forgot, that is the cytokine, and another activity is stomatal opening. But in abscisic acid, stomata closing. That is one physiological activity. After that, fall down the leaves and fruit. Promote the fall down leaves and fruit. Next one, epinasty movement, right? Epinasty movement. Next one. Hello?
Yes, sir.
Just read out the headings. I will explain.
Dormancy.
Seed dormancy removal.
Yes,
Seed dormancy removal by gibberellin. But the seed dormancy by abscisic acid. Already told you the completely opposite action of the promoters. Remember, the promoters are axins promoter, gibberellin promoter, and cytokine also promoter. The AB is the inhibitors. So in the gibberellin activity, seed germination. The opposite action done by ABA. That is seed dormancy. And ABA, another physiological activity, and seed development, I think. When we inhibit the germination, automatically embryo develop. Embryo gives the enough development, right? So that is the one physiological activity. Right, next, what is the another physiological activity?
stomatal closure.
ABA, stomata closure. Actually, stomatal opening by cytokinin. Actually, I forgot that stomatal opening by cytokinin activity. But the stomatal closing during the transpiration, when the loss of water and the high temperature, automatically inside the plant, ABA, abscisic acid released. In stress condition, mainly in unfavorable condition, stress condition, in the plant body, the produce of the abscisic acid, due to the effect of abscisic acid, automatically the stomata are closed. What happens if the stomata closed? Is there any transpiration? Is there any evaporation of water? Is it possible?
No, sir.
Hello?
No, sir.
No.
No, sir.
So that's why this is called as closing of stomata. The mainly it is formed in stress condition, unfavorable condition. Right? And next one,
Seed
another
develop-
activity.
No, sir. Seed development and maturation.
Right, seed development. Already told you, if we inhibit the germination, if we promote the germination, automatically the embryo germinate. Right? If we inhibit the germination, what happens during this time? Embryo take enough maturity to germination. That's why this is the ABA. Another importance is seed development. Seed development, nothing but here the embryo development. Right? That means the simple immature embryo, when we apply with abscisic acid, automatically it becomes mature. Right? What is the another activity?
Salt and temperature stress.
Salt and
Temperature
stress.
Yes, sir.
Right? What is the next one? No need to explain about that. It is not
Abscission.
such an important one.
Abscission.
Right? Epinasty movement, right? What happened in epinasty movement? Simple, the leaf structure. Let me share diagram. So this is epinasty. Epinasty, this just like folding. Actually, this is the leaf. But the where this leaf appear as a two edges like foldings. The mostly in epinasty movement, these edges are downward, not upward, not like upward. But this epinasty movement, the leaves like this to downward movement. So this is called epinasty. So epinasty also done by abscisic acid. Next another importance, another physiological activity. Hello?
Yes, sir.
Hello?
Yes, sir.
Clear all the abscisic acid physiological activity clear or any remaining?
Yes, sir. Salt and temperature stress is remaining.
What are the given salt and temperature? Just read out. That's not a important one, don't ignore it. Okay. Right. Next one, ethylene. Ethylene scientist. Is there given any name, scientist name? Ethylene. Adicat AB, right? Ethylene scientist, is there given or not? Okay, no problem. I don't want to waste what time and my time. Don't worry. Just read out the introduction part of the not only ethylene, all the phytohormones. Let me explain only the physiological activity because these are the very important.
Yes, sir.
Right,
So
in ethylene,
scientist name
the
is
first
given
one.
in the book.
Who is that?
R. Gane.
Gane. Okay, right.
Yes, sir.
Okay. The most important ethylene activity is from fruiting. Fruit formation, fruiting. That means the first parts convert into fruits. The effect of the ethylene, the parts convert into fruit. Ripening of fruit. Ripening of fruit, the activity done by ethylene. Right? Another next one. Triple growth, I think. Triple growth. Triple growth means gravitational growth, lateral growth, and apical growth. Where it is? In triple growth, what happened? This is the soil surface, seed. While it is germinated, the first apical growth. Apart from the apical growth, lateral growth also shows. Lateral growth and apical growth. Any obstructive shows cross growth. So this is mainly triple growth formation and fruit ripening and triple growth formation. And next one, ethylene.
Root
This is
hair
also removal
formation.
to promote the removal of leaf and fruit.
Yes, sir.
Due to the effect of the ethylene and as well as abscisic acid also, the leaves are fall down. Right? The same activity done by ethylene also. Okay? Hello? Is
Yes,
there any
Yes.
ethylene?
Yes, sir.
Ethylene clear?
Elongation growth in aquatic plants.
Elongation growth inhibitor promote.
Inhibitor, sir.
Inhibitor, right. Elongation growth done by gibberellin. Okay? That means elongation means nodes, internodes elongation. Nodes internodes elongation done by gibberellin. That complete opposite action done by ethylene. If we ethylene, that is shows it is appeared as a stunted growth, right? And what is the next one?
Root hair formation.
Hello,
Yes, sir. Root hair formation.
root
Hair formation.
root air formation.
Root hair formation.
Root air. Root air. Root air formation. Huh?
Yes, sir.
Hello.
Yes, sir.
Root air formation from stem or roots?
Extra root hair formation can be observed, sir.
By activity of ethylene. Okay. By activity of ethylene, extra root formation appeared
Yes,
in plants. Okay. But that is not an important physiological activity of the ethylene. Right? Okay. Is there any? Triple growth completed? Fruit ripening completed?
aquatic plants.
What is that? Hello?
Elongation growth in aquatic plants.
Elongation growth, I am asking you, elongation growth promoted or inhibited?
Inhibitor, sir.
Inhibitor. In aquatic plant also same, inhibitor or promoter? Inhibitor, I think.
Inhibitor, sir. Inhibitor.
Right. Already told you these are the inhibitors. Right. Is that so? Or
Any others physiological activities are there, sir?
What is that figure?
Wound healing.
Wound healing. Okay, right. Wound healing also when the plant body injured, automatically the healing process done by the ethylene activity. Okay. Right. So wound healing also another important. Here the ethylene major one, wound healing and fruit ripening. These two very important. Right. So these are the most important phytohormones. And application of these phytohormones. What are the application of ABA auxins? Auxin first the root formation during the horticulture to get number of plants. That means after we get the plants, we cut the plant and apply with IBA, indole, butyric acid and NAA. Due to the activity of IBA, NAA, the root formation arised from the stem. Right? That means if you planted the stem part, the somewhere it grows as a new plant. So that is useful in horticulture. Right? So another important of auxin, the most important in first one that and parthenocarpy. We get parthenocarpy fruit. Which is the best seed fruits and seedless fruits? Hello?
Grapes, sir. Grapes, seedless fruit.
I'm asking about the seedless or seed, which are convenient to eat.
Seedless, sir.
Seedless. That's why this is commercially very useful.
Yes, sir.
And next, gibberellin. Gibberellin, as you said, the grapes formation, seedless grapes formation, that is commercial use. Application means commercial useful to us. That is the application. Right? What is the application of cytokinin? Cytokinins, most one the delay senescence. That's why the mostly used the florists mostly use these cytokinins as the flowers appear as a fresh. That is also one commercial benefit. So mainly the most applications, auxins, gibberellin and cytokines. Is that so or any? Hello?
Sir, brassinosteroids is there, sir.
Yeah, brassinosteroids also one type of phytohormones, but no need to explain about it. Almost all the characters what we discussed so far, all the character, all the physiological activity repeated. Right? Just read out one by one headings. Brassinosteroids heading one by one. Cell division, I think something. Read out. Hello?
Growth, germination, root
Growth,
formation.
germination already we completed. We discussed. Growth, germination, both are done by gibberellin. The same activity. But here the main variation is this is the growth and regulation, growth and germination only in some particular plants, not all. Next wording.
Stress tolerance, sir.
Stress tolerance.
Yes.
Next heading. Stress
No,
tolerance, cytokinin. Sorry, ABA, abscisic acid. What happens in stress tolerance? Closing of the stomata. Next. What is another activity?
Nodulation in legumes, sir.
Nodulation in legume. Nodule formation. Hello? Root nodule formation.
Brasinosteroids increase modulation and improve nitrogen fixation in leguminous plants, sir.
Right. That is the most important character of the brasinosteroids. We know how the root formation, root nodules are formed. Already we discussed in nitrogen metabolism. Due to the activity of rhizobium bacterium, with the help of NEF genes, nitrogenase enzyme, atmospheric nitrogen fixation. During this time, brasinosteroids phytohormones released in leguminaceae plants. That is also major important physiological activity of the brasinosteroids. Next. Hello? Hello?
there is a sensation is there.
Delay senescence.
No, sensation.
What is that?
Senescence.
Repeat again.
Senescence.
Right. That means promote senescence or delay senescence. What are the given? Delay or promote? And I forgot
Promote,
one thing.
sir.
The senescence
Promote, sir. Promote.
Right. The same promote senescence done by ethylene also. Senescence promote by ethylene. Delay senescence by cytokinin. Promote senescence by ethylene. Right. As the same promotes. Is there any particular plant or common in brasinosteroids? Is there any particular plant example?
Yes, sir. Xanthium and Rumex.
Right. That's what I am asking you. Xanthium plant, the promote senescence. What is senescence? What happens if enter the senescence? The life cycle becomes end. But the green leaf turns into yellow. If it
Yes,
turns
sir.
into yellow, is this possible to prepare food material, plant body?
No, sir.
Without food material, it is goes enter the respiration process. Is it go? Is it
No,
enter
sir.
the respiration? Without a respiration,
No,
there is
sir.
a possibility production of ATP energy. No energy. Without energy, the plant can survive.
No,
Can it
sir.
survive? No.
No.
That's why that is called this process senescence. Right? Clear all?
Yes, sir.
Right. So the phytohormones completed. Clear? Next
Yes,
one,
sir.
DNA. In 7th unit, I will complete later the rest of the physiological part. That is very small lesson, small portions. I will complete next class. Let me complete the first DNA structure. RNA structure already we discussed. And DNA replication. Clear? And DNA Watson-Crick model. DNA structure important in theory and competitive level. These are all phytohormones also. The phytohormones especially just focus on physiological activity. During examination type, they may ask only physiological, write the physiological activity of auxin, write the physiological activity of gibberellin, write the physiological activity of ABA, ethylene, brasinosteroids, right? Cytokinin. And next one, DNA structure. What is DNA? Hello?
Genetic material.
Genetic material.
Material.
Just wait. With the diagram.
Nucleic acids.
Deoxyribonucleic acid. This diagram. This way, DNA, deoxyribonucleic acid, DNA structure. Mainly DNA made up of nucleotides, nitrogen bases, sugars. Let me complete. Just wait. What is the backbone of the DNA? Deoxyribonucleic acid. What is the backbone? Nucleotides. Nucleotides are the backbones. Let me share diagram. Just wait. Being downloaded. Are you able to see? Hello?
Yes, sir. Yes, sir.
Sir.
Yes.
Right. So the mainly it is made up of nucleotide. Nucleotide appear as a polymer. So nucleotide, this is the total, this called as nucleotide. If you separate nucleotide, nucleoside, if you separate the nucleoside, nitrogen bases, right? So first nucleotide, in nucleotide mainly phosphorus, sugar, nitrogen bases. These three together form as a nucleotide. Like this number of nucleotide form as a DNA structure. This is one nucleotide. This is second, third, fourth, like this number of nucleotides. These are the considered as the backbone of the DNA, right? So in nucleotide, phosphoric acid, P for phosphoric acid, sugar, deoxyribose sugar, nitrogen bases, right? So next one, if we separate the nucleotide, the nucleosides, in nucleosides, only without phosphorus, sugar and nitrogen bases, these two considered as a nucleoside, right? If we separate the nucleoside, only nitrogen bases are formed. What are the nitrogen bases? Hello? Hello? What are the nitrogen bases in DNA? Right? Adenine, thymine, guanine, cytosine.
Cytosine.
This is one end. The same copy to another strand. Already told you the DNA anti-parallel structure. These are anti-parallel 5 end to 3 end, 3 end to 5 end, anti-parallel structure. And it has complementary sequences. So for example, one end adenine, automatically another end thymine. One end guanine, automatically another end cytosine. So remember these nitrogen bases bond with hydrogens. Always the guanines and cytosines, triple bond, adenine thymine, double bond. So these nitrogen bases mainly two types, purine and another one is pyrimidine. Purines and pyrimidines. What are the purines in this? Purines A, G, adenine, guanine, purines. Pyrimidines are TC, thymine, cytosines are pyrimidines, right? Hello?
Yes, sir. Yes, sir.
Right. Now, another important one is here so bond formation between in nucleotide sugar to phosphoric acid diester bonds formed. Phosphodiester bonds formed. What happened? How the formation diester bond? Let's see. So here the phosphate phosphoric acid bond sugar. Same sugar. So this two ester bond. This is ester bond. This is also ester bond. One ester bond sugar 5 prime end. One ester bond with 3 prime. 3 end or 3 prime. Another ester bond 5 prime. So this diester bond always formed with sugar. Sugar here made up of 5 carbon sugar. In 5 carbon sugar, the one ester bond formation covalently with 5th carbon and another bond formation with 3rd carbon. So this is diester formation. The important nucleoside glycosidic bond formed. This glycosidic bond formation between sugar molecule and nitrogen bases. Sugar and nitrogen bases, the bond formation glycosidic bond. So this is DNA structure. In this, hello, 3 and sorry, opposite 5 and 3 end. Is it clear? Hello?
Yes, sir. Yes, sir.
Right. About it, you can easily understand DNA Watson model. In DNA structure, these are the nitrogen bases. Nucleoside with diester phosphate. Nucleoside nitrogen. Enough. Right, clear? Hello? Hello?
Your
Yes,
voice
sir.
is cutting, sir.
Sir, your voice is not clear.
Some
Hello? Is it clear?
No, sir, we are not audible.
No, sir.
Hello?
Yes, sir.
Now clear.
Is it okay?
Okay, now.
Yes, sir. Okay.
Okay, now.
Right. Now let's see Watson and this Watson-Crick model. In Watson-Crick model, in DNA double helix, 2 grooves are formed. This is major groove. This is major groove. This is minor groove. 2 groove form as a pitch. Minor, major. These two constitutively form as a pitch. One helix. The length of the helix 3.4 nanometer. 3.4 nanometer. In 3.4 nanometer, the nitrogen bases pair appeared in 10. 10 nitrogen bases appear. Right? 10 nitrogen bases appeared, and the radial this bit, the length of these two strands 20 angstroms. This end and this end length 20 angstroms. And always the purine pyramid appear in 1:1 ratio. Guanine, cytosine, adenine, thymine, purine, pyrimidine. So these two pyrimidine, right? These two purines. Always the purine equal to the pyrimidine. That is cos one. This is the 1:1 ratio between purines and pyrimidines, right? Right, major group, minor group, and between the nitrogen bases, hydrogen bonds are formed. Already told you the guanine cytosine 3 bond, adenine thymine double bond, thymine adenine also double bond, cytosine guanine also double bond, right? Is it clear?
Yes,
This
sir.
1:1 ratio called as Chargaff rule, because purine equal to pyrimidines. That is called this rule called as Chargaff rule. The first one is a double helix, always right angle. The folding right angle, unable to see in this online classes. Always one fold. Always this is, for example, this is the axis. For this axis, the DNA stand right angle, folded at right angle only, right? So in this, the pitches are, one is minor groove, another is major groove. These two constantly called as the length of this groove 3.4 nanometers. And next one, this terms minor major called as pitch. A pitch constantly formed as a constantly formed. Just wait. Constantly formed as a pitch. Okay. Watson-Crick model. Watson-Crick model important in short answers. DNA structure important in short answer, and also useful, important in competitive level also. Right? For example, I want to ask a simple question to you. If the pitch length 340, pitch length, DNA pitch length 340 angstroms, that how many nitrogen bases present in this pitch? Hello? Or 34, sorry.
ଯାଉ ସରିଲା ଆଉ ଟିକେ ତୋର ନାଇଁ ଜିମି ଆଉ କି ଭାବୁଛୁ ମୁଁ କହିଦଉଛି ଭାଉଜଙ୍କୁ କହିଦେ ଟିକେ
34 angstrom length. In 34 angstrom length, how many? In 34 angstrom length, how many nitrogen bases are present? Hello? Three power 10. In 34, how many?
100, sir.
That's it. Discuss about then. DNA replication. So what is DNA replication first? Do you have any idea?
Can you repeat that, sir?
DNA replication. What is meaning of replication?
Formation of a new
The
strand.
two double.
That's it. Formation of one DNA strand, mother strand, template strand, formation new DNA strand. That is called DNA replication. What are the requirements for DNA replication? What are the requirement? DNA template strand, right?
Sir, yes, sir.
DNA template strand. Hello? DNA
Yes, sir.
template, DNA polymerase, primers, single stranded binding proteins, helicase, they are all most important, most requirement of the DNA polymerase, right? Is it clear? DNA structure clear before that? So this is a
Okay,
complex
sir.
process that takes place during cell division. Mainly the DNA replication takes place during cell division process, especially in interphase S phase. What happens in interphase S phase? DNA, for example, this is the cell. Are you able to see this diagram?
Yes, sir.
Okay. Unable to draw this diagram. Actually, it's a low battery. So anyway, up to this bracket battery down, just listen. So during the cell division, interphase S phase, synthetic phase. During synthetic phase, one DNA strand become double, right? That is called replication. So replication is a semi-conservative process. Why it is called semi-conservative? Semi-conservative means actually first double stranded DNA, right? Just imagine, just here the unable to draw this due to the poor battery, unable to draw the diagrams. Just imagine, actually the DNA is double helix, right? Hello?
Yes,
Yes,
sir.
Hello?
Yes, sir.
Double helix DNA.
Yes, sir.
Each one in double helix, each one can prepare newly synthesized DNA, right? After prepared DNA daughter strand, after synthesis, later this old DNA and newly synthesized DNA, the both are coiled and form as a double helix DNA. That's why it's semi-conservative. Means in semi-conservative, one is new strand, one is old strand. Are able to understand? Hello? In
Yes,
semi-conservative
yes, sir.
means first of actually this is the low battery, but why it is not showing? First of all, are you able to see this diagram? Okay.
Yes, sir. Yes.
Yes, sir, we
Hello?
are able to see it.
Right. You can observe the blue here the black line and blue line. Black and blue line. Okay. Are you
Yes,
able
sir.
to see black line first? Like a ladder. Black and opposite complement
Yes,
is
sir.
blue, right? Here the
Yes.
two blue lines are new stands. Two black lines are old stands. After synthesis, one blue line, one black line together form as a helix and deployed that is double helix DNA stand. In this DNA stand, one is new. Are you clear my voice? Is it clear? Hello?
Yes, sir.
Now clear, sir.
Clear, right? Are you clear with semi-conservative process? Simple and semi-conservative process after synthesis.
Yes, sir.
Okay,
One
just
example,
let me clear
no
one
stand
simple
and
doubt. Double helix DNA. Let me ask a simple question. In double helix DNA, how many strands are present?
2 strands, sir.
4 stand.
2, sir.
Double 2.
Hello? Hello?
Yes, sir.
No?
Yes, sir.
Right. In double helix DNA, how many DNA strands are present?
2 strands,
2
sir.
Two
stand.
strands, sir.
Two strands. After synthesis, how it becomes? After replication, these two strands becomes
2.
3, 4.
how much?
Again, 2
strand.
strands of DNA will be produced. From leading
4.
strand, 1 strand of DNA is synthesized. From that strand, again another strand of DNA is synthesized.
Hello?
Yes, sir.
Some network problem here. I'm in village. Now clear? Now clear? My voice is clear?
Yes, sir. Now it is clear.
Right. After replication, two strands become 4, right? Hello?
Yes, sir.
In this 4, later convert into two DNA strand separate and two DNA strand separate. In two DNA strand, one is old one is new one. And remaining two, one is new one and one is old one. So this is called as semi-conservative method, right? Clear? Now let's see the requirement. I already
Yes,
told
sir.
you the requirement for the DNA polymerase, DNA primers and SSB single standard binding proteins.
Heritage.
Single standard binding protein, right? Are able to see the diagram? Now let's see the mechanism.
No, sir.
The simple, there are three steps during DNA replication. One is opening of double stranded helical structure DNA, attached priming to the template strand. Attached of newly DNA. Attached of newly DNA polymerase. During separation of DNA, the two strands uncoil at the specific site known as origin. Let me show diagram. So where the replication fork started, that place called as origin. The place called as a origin, right? At the end of the process, DNA polymerase enzyme start the origins and assemblage. First of all, actually, what happened? Before that, let discuss about one by one function. What is the function of DNA polymerase? As the name given polymerase, it is synthesize the new DNA. And next one, and another one, helicase enzyme. The enzyme, this enzyme that involved in the unwinding. Actually, what is the nature of DNA? DNA always supercoiled. Supercoiled nature, to synthesize the DNA strand, it should be unwound. This unwinding activity done by helicase enzyme. Is it possible if the DNA structure in supercoiled nature, winding nature, can it synthesize new DNA? Hello? Hello?
No.
Yes,
No,
sir.
sir.
If the DNA strand supercoiled nature, coiling nature, is it possible to synthesize new DNA?
No,
sir.
sir.
Almost is winding nature, there is no possibility to finding DNA strand. That's why with help of using helicase enzyme, this enzyme unwinding the double strand, helix, DNA helix unwinding. That is the first function of this helicase. After unwinding with help of helicase, here the single stranded binding protein, SSB. We can observe, actually this marker not shown